Learning outcomes. After studying this module, you shall be able to describe what is meant by genetics, outline using appropriate examples the laws of genetic inheritance, describe the ways in which genes influence human development and behavior, explain using experimental examples what is meant by heritability. Introduction. Give me a dozen healthy infants, well informed and my own specific world to bring them up in, and I'll guarantee to take any one at random and train him to become any type of specialist I might select. A doctor, lawyer, artist, merchant chief, and yes, even into beggarman and thief, regardless of his talents, penchants, tendencies, abilities, vocations, and race of his ancestors. Contrary to the viewpoint proposed by the famous behaviorist John B. Watson, who uses nurture, which is environment, to explain our behavioral processes, researchers in the area of behavioral genetics claim that different aspects of our behavior, including our consciousness, result from a complex interplay of nature and nurture. While most researchers today do not question the role of genetic influences on psychological traits, most studies focus on how much influence there is and how genes work to shape the mind. The field of behavior genetic studies human twins and adoptees to uncover both environmental and genetic influences and allows us to estimate the heritability of various traits. Genomes and introduction. In 1953, James Watson with Francis Crick described the double helix structure of deoxyribonucleic acid DNA, the chemical compound which carries the genetic instructions for building, running and maintaining living organisms. Over the 1970s, methods were developed to determine the order or sequence of the chemical letters in DNA. The National Institute of Health (NIH) and the Department of Energy in the USA collaborated with international partners to completely map and understand all the genes of human beings, together known as the human genome. This concerted public effort was the Human Genome Project. In April 2003, researchers working on the project made headlines by successfully completing the Human Genome Project under budget and more than 2 years ahead of schedule. While the scientists have figured out the sequencing of the different DNA molecules, they still need to identify the functions associated with these molecules in terms of mind, body and behavior. The human genome contains between 30,000 to 40,000 genes, segments of DNA that serve as basic units of heredity. Genes working in complex combinations and together with forces in the environment become blueprints that shape development and all basic bodily processes. These genes are located on chromosomes, thread-like structures found in the nuclei of cells. Each human cell consists of 23 pairs of chromosomes. Chromosomes are composed of DNA, deoxyribonucleic acid. Most cells in the body contain 46 pairs of chromosomes, existing in pairs, one set of chromosomes from father and the other set by the mother. All normal developing humans have the same characteristics, 2 eyes, 1 nose, etc., as most of the genes within the human genome are same for most individuals. Only a small number of the genes vary across individuals, which results in differences like different eye color, hair type, height, etc. Genetic similarities also explain why persons who are related resemble one another more than persons who are totally unrelated. The closer the familial ties between the individuals, greater the proportion of chromosomes and genes shared, which in turn results in greater physical similarity. However, the effect of genetic similarities is not restricted to physical appearance. The exciting question here is to understand the role of genetics in determining personality. Controversy about genes and personality. On January 2, 1996, the New York Times published an article about how scientists had discovered a single gene which could be used to explain novelty-seeking behavior. Not surprisingly, the article, like many other researchers done in the area of behavior genetics, created a lot of stir. Popular media sources also tend to sensationalize and present a simplistic account of the work done in the area. However, scientific initiatives like Human Genome Project, which aim to map genetics to explain a range of personality factors, have re-linked interest in the area of behavior genetics. Modern behavioral genetics who study personality tend to be very careful about addressing the implications of work as findings from these studies can be misused for the propagation of political agendas. For examples, advances in the area which helps in pinpointing genes responsible for delinquent behavior will cast a shadow over the possible positive effect of reformation programs for these children. Another ideological concern comes from the idea of eugenics. Aimed at improving the genetic composition of human race, eugenics movement historically advocated selective breeding to achieve these goals. The term was first studied by Sir Francis Galton, a respected British scholar and cousin of Charles Darwin, who believed that the human race could benefit by selectively breeding individuals who have desired traits. In many other countries, the movement took sinister turns, with eugenetics in the US attempting to stop the transmission of negative or undesirable traits from generation to generation by sterilizing unfit individuals. While the first sterilization efforts were targeted exclusively at mentally ill people, exclusively traits like alcoholism, criminality, chronic poverty, blindness, deafness, feeble-mindedness, and promiscuity were also deemed serious enough to be considered for sterilization. Many people in the society are therefore concerned about the misuse of findings from behavior genetics to advocate selective breeding for creation of a master race. This explains the care and caution employed by behavior genetics in explaining their findings, its uses and potential misuses. Additionally, genetics also claim that finding a personality trait that has a genetic component does not mean the environment is powerless to modify the trait. Goals of behavior genetics. One of the primary goals of behavior genetics is to determine the percentage of individual differences in a trait that can be attributed to genetic differences and the percentage that can be attributed to environmental differences. These individual differences, while influenced by genetics, are not determined by genetics alone. Behavior genetics aims to understand what percentage of individual differences can be attributed to genetics and what percentage can be attributed to the environment. While genetic research attributes as much as 90% to genetic factors to explain physical attributes like height, the effect of environmental factors cannot be ignored. For example, the average adult height in the US has increased by 2 feet over the past 1 century, likely to the increase in the nutritional value of the food consumed. Behavioral genetics also aims to determine the ways in which genes and environment interact and correlate with each other to produce individual differences. Precise identification of the factors in the environment which influence personality, example, socialization by parents, community, school, teaching style, etc., is another aim of behavior genetics. Heritability. Heritability is a statistic that refers to the proportion of observed variance in a group of individuals that can be accounted for by genetics. It describes the degree to which the genetic differences between individuals cause differences in an observed property such as height, extraversion or thrill seeking. Statistically, heritability is defined as the proportion of phenotypic variance that is attributable to genetic variance, wherein phenotypic variance refers to the observable individual differences such as height, weight, etc., while genotypic variance refers to the individual differences in total collection of the genes possessed by each individual. As heritability is a proportion, its numeric value will range from 0.0, wherein genes do not contribute at all to the phenotypic individual differences, to 1.0, wherein genes are the only reason for the individual differences. For human behavior, almost all estimates of heritability are in the moderate range of 0.30 to 0.60. The quantity 1.0 heritability gives the environmentality of the trait, that is, the percentage of the observed individual difference that can be attributed to the environment. The greater the environmentality, smaller the heritability and vice versa. If the heritability of the human behavior 0.30, then the environmentability of the human behavior will be around 0.70. Misconceptions about heritability. Heritability is one of the most misunderstood concepts in psychology. One of the biggest misconceptions is that heritability can refer to a single individual. In reality, the roles of genes and environment in influencing the individual is inextricably linked. Both these factors play a role in determining physical and psychological characteristics. Heritability therefore refers to differences in sample of population and not just an individual. For example, heritability of 0.30 informs us that on average about 30% of the individual difference that we observe in, say, gregariousness may in some way be attributable to genetic individual difference. It does not, however, mean that 30% of any person's gregariousness is due to his or her genes and the other 70% is due to his or her environment. The second misconception revolves around the consistency of heritability across environments. In fact, heritability depends on the range of typical environments in the population being studied. If the environment of the population is fairly uniform, then heritability may be high. But if the range of environmental differences is very large, then heritability may be low. The final conception is around heritability being a precise statistic. Heritability at best is an estimate of the percentage of phenotypic differences due to the genetic differences. Errors, unreliability of measurements, fluctuation due to use of correlation with other different samples results in distortion of heritability statistics. Therefore, we can say that heritability statistics is not precise, fixed across environments or referring to any one individual. Behavioral genetics methods. The field of behavioral genetics has an enormous potential to uncover the genetic and environmental influences on behavior. These methods are based on solid foundation of theories and methods which have successfully delineated elements that contribute to complex behavioral characteristics. Some of the traditional methods for these include family studies, twin studies and adoption studies. Selective breeding with animals is another method. Family studies. Family studies revolve around the correlation of the degree of genetic overlap among family members with the degree of similarity in personality types. While parents do not share a similar genetic pool, they share 50% of their genes with each of their children. If a trait is highly heritable, family members with greater genetic relatedness should be more similar to one another on the trait than family members who are less closely genetically related. However, the problem arises when members of a family who share the same genes also usually share the same environment. This confounds the genetic influences with environmental influences. Thus, family studies are never definitive. Twin studies. Twin studies, with their attempt to disentangle the environmental and genetic backgrounds of a large amount of traits, are one of the favorite tools used by behavior genetics. Twin studies estimate heritability by evaluating whether identical monozygotic twins share 100% of genes, more similar than fraternal dizygotic twins who share only 50% of genes. If monozygotic twins are similar than dizygotic twins, this provides evidence of heritability. Twin studies methods rests on two assumptions. The first assumption is the equal environments assumptions, which assumes that fraternal and identical twins raised in the same homes experience equally similar environments. But some research suggests that parents, teachers, peers and other may treat identical twins more similarly than fraternal twins. The second assumption revolves around representativeness of the general population from which they come. Generalizations about heritability based on twin studies might not be representative about the general population. Despite these limitations, the value of twin studies research in studying heritability cannot be negated. Adoption studies. Adoption studies based on positive correlation of traits between adopted children and adoptive parents provide evidence of environmental influence. The evidence on genetic influence is provided by positive correlation between adopted children and genetic parents. Adoption studies solve for the limitation arising out of equal environments assumptions, as genetic and environmental causes are unconfounded here. However, the assumptions that adopted children and their adoptive and genetic parents are representative of the general population is questionable. Also, if the children are placed with adoptive parents who are similar to their biological parents, the correlations are artificially inflated. Selective breeding. Selective breeding entails the process of identifying animals with certain desirable characteristics and having them mate with only those animals who have similar characteristics. However, this method is useful only when a desired trait is heritable. While selective breeding studies cannot be ethically conducted with humans, selective breeding studies of dogs are quite common to study the effect of heritability. This includes not only the study of heritable observable qualities such as physical traits, example, wrinkled skin, eye color, etc., but also behavioral characteristics such as sociability, agreeableness. Studies conducted over years, Scott and Fuller in 1965 have suggested that personality characteristics are not just physical characteristics, but it can be inherited. However, heritability of behavior traits in dogs cannot help us to transfer the same conclusions about heritability of personality characteristics in human beings. Major findings from behavioral genetic research and molecular genetic research. From thousands of different studies using many different methodologies, we have uncovered various influences of genetics on different aspects of our personality. Personality types. A review of literature on more than 25,000 twins by Henderson in 1982 has found substantial heritability for personality traits of extraversion and neuroticism. One of the recent large-scale twin studies in Australia found heritability for neuroticism to be 47%. The findings from adoption studies for extraversion and heritability suggest modest heritabilities. A study by Pedersen in 1993 suggests heritability estimates based on adoption studies to be around 40% for extraversion and 30% for neuroticism. Activity levels also show a moderate heritability of 0.40, suggesting a moderate role of genetic differences in the energy expended by individuals. One of the most interesting studies in this area is the Minnesota Twin Registry, which explored the heritability of psychopathic personality characteristics like cold-heartedness, callous emotional style, malevolent, enjoying manipulating others, etc. Most of these personality traits showed high to moderate heritability. Attitudes and behavior. Attitudes, with their stability over time and influence on behaviors, are often regarded as a part of one's personality. Minnesota Twin Register reports a heritability of 0.63 for traditionalism, attitudes favoring conservative over modern values. In another longitudinal study done with over 654 participants in Colorado, USA, it was found that genetic influences on attitudes are observable since 12 years of age. Genetics also plays a role in your occupation-related preferences. A study by Lee, Ellis, Bonnen and Stephan in 2003 approach taken involved comparing 435 adopted and 10,880 genetic offsprings regarding various aspects of the jobs they prefer, along with the 7 different measures of parental social status. In line with the hypothesis that genes influence both occupational related preferences and social status, higher average correlations were reported between several of these preferences and the parental status of the genetic offspring than for the adoptive offspring. Smoking and drinking. Genetics also influence a person's risk of becoming a smoker as well as the risk of alcohol dependence. Finding from a 1981 Australian twin panel cohort confirm a significant positive genetic correlation between regular smoking and the risk of alcohol dependence. A bivariate genetic analysis study on tobacco smoking and alcohol drinking in China also suggested a common genetic vulnerability to tobacco and alcohol use in male twins. Despite the evidence for influence of genetic factors, the same studies that suggest moderate heritability also provide good evidence for the importance of environmental influences. For example, personality characteristics show heritabilities in 30 to 50%, range from point towards substantial degree of environmentality in 50 to 70%. Molecular genetics. Molecular genetics refers to the technique designed to identify specific genes associated with personality traits. The most common method, called the association method, attempts to identify whether individuals with a particular gene have a higher or lower score on a personality trait than individuals who do not possess those genes. Research in this area has been pretty nascent, with the first study getting published only in the 1996 by Benjamin et al. In 1996. Most frequently studied allele in this regard has been the D4DR gene located on the short arm of chromosome 11 and acts as the dopamine receptor. In studies examining the association between D4DR gene and a personality trait involving novelty seeking individuals with the long repeat version of D4DR gene were found to be higher on novelty seeking than individual with the short repeat version of gene. Although this association has been replicated several times, there have also been multiple failures to replicate association, and even when replicated, the association has been weak. As such, it is important to exercise caution while interpreting the results of molecular genetics. Genes and the environment. The relationship between genes and environment is not a simple linear relationship. Rather, the different ways to conceptualize the ultimate effect of gene-environment interactions on the expression of a trait or behavior makes the issue more complicated. These complex forms of genetic behavioral analysis include genotype-environment interaction and genotype-environment correlation. Genotype-environment interaction. It refers to the differential response of individuals with different genotypes of the same environments. A specific aspect of the environment might have a greater effect on some genotypes over others. For example, introverts tend to perform better on cognitive tasks in environments with little stimulation, while extroverts might make more errors when the tasks are boring or monotonous. On the other hand, introverts will perform poorly as compared to extroverts when they are distractions like noise and other disturbances in the room. Genotype environment correlation. The genotype environment correlation occurs when the genotypes are selectively exposed to different environments. Here, individuals with the different genotypes are exposed to different environments. For example, parents are more likely to encourage athletically inclined children to athletics than athletically disinclined children. Plomin, De Vries and Liohlin in 1977 refers to three types of genetic environment correlation. 1. Passive genotype environment correlation, which refers to the association between the inherited genotype of an individual and the environment in which they are raised. For example, parents who are verbally inclined may pass on genes which make their children verbally inclined. While the parents might make books available in the environment, the child has done nothing to cause the books to be there. 2. Reactive genotype environment correlations refers to the association between an individual's genetically influenced behavior and the influence of others' reactions to that behavior. For example, a mother might hug a cuddler more than a non-cuddler because the cuddler responds more positively than a non-cuddler to the mother's cuddling behavior. 3. Active genotype environment correlation, which refers to the association between an individual's genetic propensities and the environment selected by the individual. For example, an extrovert might seek out very different social environments than those who are shy and withdrawn. These genotype environment correlations can have a positive and negative implication. For example, it can explain why individuals who have a genetic propensity to engage in sensation seeking behaviors affiliate with drug abusing peers. Summary. Behavioral genetics attempt to determine the degree to which the individual differences in personality are caused by genetic and environmental differences. Heritability is the proportion of observed variance in group of individuals that can be explained or accounted for by the genetic variance. Environmentality is the proportion of observed variance in group of individuals attributable to environmental variance. Over the past few decades, we have gathered more compelling evidence about influence of heritability and environmentality of personality. Findings from the 4 behavior genetic methods, selective breeding, family studies, twin studies and adoption studies have helped us to strengthen this case. Research also suggests a moderate role for heritability on personality variables such as extroversion and neuroticism, as well as attitudes and behavior around drinking and smoking. The same studies also make a case for the same variables having a moderate to strong environmentality. Some behavior genetics also examine genotype environment interaction and genotype environment correlation, which opens us new and interesting avenues for this field of research.